Okay, 안녕하세요. Hi everyone. I'm Hoi Lok. You can call me Cole. So I'm the CDMO application scientist from VectorBuilder. So thank you for BRIC for hosting this webinar. So today the topic will be about reinforcing and accelerating the development of cell and gene therapy, which is also called CGT, with end-to-end CDMO service. Let's begin. So in today's webinar, I'm going to talk about a very brief introduction and background of VectorBuilder. And of course, I'll introduce the current CGT landscape and some bottlenecks of the development of such kind of drugs. And of course, to support CGT industry to develop, to accelerate, VectorBuilder full spectrum solutions will help researchers, drug developers to have better access to different tools and better service to optimize their drug design. And of course, I'm going to give you a few case studies on how a CDMO, or maybe not just CDMO, like companies like VectorBuilder, service provider, how can we develop CGT together with drug developer? And at the end, I will talk about our CDMO platform and our end-to-end service to accelerate your pipeline. So first of all, about VectorBuilder. So first of all, what is gene delivery? So when we talk about gene delivery, usually it refers to a process to introduce a foreign gene into your target cells or target tissue by a lot of different gene vectors, such as biological vector, for example, viral vector, chemical or physical methods, and recently, using LNP, lipid nanoparticle, to deliver mRNA is also very popular after the COVID-19 situation, pandemic. And there are actually a lot of ways to deliver gene. And, after you can deliver the gene, you enable a lot of different research areas, such as life science, genetic medicine, agriculture and food, green energy, and even industrial fermentation. So VectorBuilder, our core concept is to support gene delivery revolution. And since 2014, we have been expanding our business and our team, our facility, to really catch up with the entire industry around the world, to support researchers from different kind of institute and company. And right now we have been expanding our office in every corner around the world. We have been expanding our GMP facility. And last year, fortunately, we have become a unicorn company. And we promise that we'll be providing better service to everyone who is working really hard on gene delivery. So here are the 4 major offerings from VectorBuilder. First of all, there will be research vector cloning and virus packagings. For virus, typical one like AAV, adeno-associated virus, or lentivirus, retrovirus. And recently, we have also added on a lot of different kind of viral vector to our service list as well. Then the second one will be our gene delivery CRO service. So for CRO, usually we're talking about a lot of, for example, library construction or screening, evolution kind of service. So one of our very famous service is actually capsid evolution for AAV to enable better efficiency for gene delivery. Then the third one will be our genetic medicine CDMO, and you may also refer to GMP manufacturing of your drug product. Then the last one will be intellectual property out-licensing, which will be online very soon. That will be about our, for example, some novel new AAV capsid promoter that VectorBuilder have engineered. So currently, with our world's first gene vector Amazon platform, we really expand our reach to all around the world. We have served more than 90 countries. And right now, to better support our VectorBuilder community, we have set up more than 10 branches and offices. And here is some simple statistics of our company. We have more than 700 employees worldwide to support customer that comes from different country, different culture, who speak different language. And actually, more than 25% of us hold a postgraduate degree, so we can better understand your issue, your problem. We can help you to troubleshoot. And as a bioscience, biotechnology company, we need to keep evolving and developing our own technology. That's why we spend about 15 to 20% annual budget on R&D projects. And currently, we have more than 600 ongoing R&D projects inside the company. And on the other hand, for our manufacturing facility, the total area already reaches 15,000 square meters, and we have more than 10 GMP suites. Okay, so after I talk about our company, so let me introduce a little bit more on the cell and gene therapy industry. What are the problems that the whole community is facing? So first of all, why do we want to do cell and gene therapy? Why do we want to do gene delivery? So there are a lot of disease, rare disease, such as the case study here, Mankis syndrome, that people actually suffer from some inherited situation, maybe with a copy of gene that is not functional with, for example, mutation, deletion on it. So by delivering a functional replacement of this gene, you actually can change this person's life by taking off, continuous use of some medicine, for example. So this current case study is a real life, case study of how we help a biopharma called Lantu to help saving life. We provide CRO and CDMO service for this world's first AAV-based gene therapy for Mankis syndrome. And this kid, it's the first patient in the world who actually received this drug. And luckily, after the receiving of this AAV-based drug, he's still surviving very well right now. And to quote our chief scientist, Ruslan, he said that it is actually a very intense race to develop such GMP AAV drug. And, to race against time and actually optimizing a large size cargo for the DNA genome inside the AAV particle is actually very challenging, but also, of course, it's very satisfactory for us. So to deal with such kind of rare disease, to solve a lot of unmet medical needs in the world, the gene delivery market is actually growing in a tremendous way. The current market size is about 15 billion US dollars per year. But of course, we projected by 2028, it will actually become 35 billion USD per year, which includes research and development like R&D, vector, virus vector or non-virus vector preparation, gene delivery, CRO, and also the CDMO, the manufacturing of this kind of drug. So now let's take a look at the approved CGT drugs. So for in vivo cell and gene delivery, which means you are using, for example, a viral vector to deliver a piece of functional gene into your patient directly, for a direct injection, local administration. There are several options that you can actually pick. For example, the most popular one recently is adeno-associated virus, AAV. And of course, there is also others viral vector of choice, for example, adenovirus, HSV, VSV, so on and so forth. But of course, recently, you can see that more and more AAV vector that have been approved by, for example, FDA and EMA, by a lot of regulatory agencies around the world because of its safety, lower risk of use of such kind of AAV vector. One advantage about AAV is that the gene is not going to integrate into your chromosome, which is a very huge advantage when you want to get the approval from your regulatory agency. Then next, if we look at the ex vivo cell and gene therapy, which means usually it's a modified, a gene modified cell therapy, for example, CAR-T. CAR-T therapy, it's by integrating chimeric antigen receptor into your immune cells, particularly T cells. And after the modification and certain steps of cell manufacturing, then it is put back onto your patients to help fighting your cancer. So such kind of ex vivo CGT actually is also very popular, and a lot of the ex vivo therapy that is shown on the screen here, actually half of them are CAR-T, and the other half are modified cells that is to deal with other kinds of rare disease. So to quote the data from ASGCT, we have been seeing more and more clinical pipeline of such kind of cell and gene therapy getting into clinical phase. And of course, eventually they will get approved for marketing. So if you compare in vivo and ex vivo therapy, you can see that actually this year, we are seeing a lot more in vivo delivery of gene by using, for example, AAV. And for ex vivo cell therapy, as I say, almost half of them are CAR-T, which also matches what we see in the approved ex vivo therapy. So if you look at the global status of the clinical development of such kind of CGT, you can see that in preclinical stage, there are a lot of people actually working on it, almost like 1,500 research that is ongoing. However, you want to get enough evidence that your drug is safe, is effective, and you want to apply it on human, you need to get approval from the regulatory agency before you actually enter the clinical phase 1, 2, and 3. You can see that the number actually keep dropping. And finally, the one that actually goes to the market that can be commercialized is just, one of 100, really difficult to get to the final stage. So why is that? What is the bottleneck of developing such CGT drug? So from our perspective, there are two main reasons. The first of all, the design of such kind of gene delivery vector. So for the design, first of all, the very first bottleneck you will meet is your do-it-yourself vector preparation when you are doing drug discovery. It really takes a long time. It's actually quite challenging for preparing such a good quality viral vector, for example. Then next, your not every lab is equipped with, for example, animal models or library construction, or even using AI or using machine learning to facilitate how you analyze the data, how you build such a library. So lack of screening and verifying strategy is actually one of the bottlenecks, which might hinder the development of such kind of CGT. Then, of course, if you finally find a drug candidate, you enter into preclinical phase, you put it on different animal models, you try to test the efficacy and safety, whether it's actually good enough for clinical, whether you have the resources or not. Actually the animal model could be quite expensive, so that is also one bottleneck. On the other hand, you are trying to prepare for your GMP grade, your clinical grade CGT drug. So you have to start developing your manufacturing process. So actually there are tons of choices that you can pick to manufacturing such a drug. Without much experience, actually a lot of people get lost in this procedure. There are too many ways you can operate. How do you get the best quality attributes for your CGT drug? So that is one problem. And later, when you finally get into the clinical stage, when you are applying it on human, you need your GMP grade virus vector, plasmid, mRNA, LNP. So your manufacturing becomes so important that you don't want to see any failure. However, failure always happens. One step in your manufacturing goes wrong, you get a really bad quality attribute, and then it's not safe enough. You cannot, get approval from the regulatory agency to put such kind of drug on human. It's just not ethical. So manufacturing failure is also a big problem for drug developer. And of course, during your clinical phase, safety, efficacy issue is also blocking a lot of people's way to get into phase 3 and commercializing their drug. So for manufacturing, let me expand a little bit for the development of such kind of manufacturing process. What we imagine is that after I have been through so many experiment research, I finally find a very good drug candidate. I just paid someone or I just build a facility and then manufacture the drug. Is it that simple? So what we imagine is that, okay, we develop such a process to manufacturing this drug, and then we do the GMP batch. We manufacture it, to have the CMC document ready, submit it to the regulatory agency, and then done, we apply it on human. That's what we imagine. But what actually might happen in a lot of drug pipeline development that we have been seeing is the failure to address the challenge in such kind of process when it is still in small scale, in lab scale. It's actually very easy to control a lot of parameters and conditions, even for contamination. But once you want to scale it up, for example, for human use, you need a higher dosage than your mouse model. In this case, with such big manufacturing campaign, a lot of accidents can happen. A lot of, for example, purification can go wrong, that you have a very low yield of your drug. So the development of such kind of GMP manufacturing process can become very, very long, and you have to keep repeating and optimizing, and then it already, takes 1 or 2 years before your GMP batch. And because of such kind of failure, maybe it will be, kind of continue to your real GMP batch manufacturing where you fail to meet your desired quality. So you have to keep doing change. And this GMP batch, it's a waste of time and money. So let me introduce VectorBuilder's full spectrum solutions. Like, how can VectorBuilder solution facilitate your development of CGT drug? So first of all, for Vector DIY, the very first step in your drug discovery, a lot of researchers actually talk to us in the very beginning. They have been finding it very hard to, describe a vector to their supervisor, and to design such a vector. How do they communicate with their coworkers? How do they troubleshoot some problem? How do they, when they want to optimize the vector? And also, it's actually quite tedious, and sometimes it could even be challenging to produce such vector. So VectorBuilder actually have an online vector design studio, which we build it into a way that is user-friendly, is Lego-like. So it's just drag and drop, and then you can finish the design and assembly of your vector. And also we have a very, sophisticated presentation of this vector, so that, for efficiency information processing. To match this demand of such kind of DIY vectors, we also build a scalable Industry 4.0 production model, so that when our customer designs something on our studio, we have our price and information shown immediately for you to assess, like, how it would look like if you want to order it from VectorBuilder. So with such kind of model, this is a one tool for all. So we want to revolutionize the whole gene delivery R&D process with you. And currently, we have more than 700 vector systems. With this tool, you can do intuitive design with our components library containing more than 400,000 different components that we have accumulated in our database. And of course, our price is very and highly affordable, and also rapid turnaround time, so that when you order it, we really deliver it quick enough. You can test it in no time. So this is our growing workforce that, each year we have been providing more than 250,000 vector design. And then for vector cloning projects, there are over 80,000 vectors per year. Virus packaging, 40,000 vectors per year. And for plasmid DNA preparation, 60,000 vectors per year. So now you can see actually how many people are trying to get away from doing the cloning or virus preparation themselves. So of course, for VectorBuilder, we don't want to just stop here, providing research-grade vector, because throughout these years, we have built up a lot of experience with virus vector cloning, and the behavior, the properties of these vectors. So we call it the know-how of this vector. So we want to transform it into, for example, first of all, the high-value CRO service. We know how to do screening in vitro and in vivo, and also we know how to build a high-quality library so that it enables us to provide capsid evolution, promoter screening, such kind of service. And please talk to us for more evolution or screening strategy later. Then next, we transform our know-how into CDMO. So with the technical know-how that I just described, we build up an integrated platform, for example, for process, for the manufacturing, in-house QC panel, professional process development team, and our advanced technology. We try to bring the cost and turnaround time down for your GMP manufacturing campaign. So by providing such a GMP platform solutions, we want the drug developers out there to know that if you can pick a good platform, what you imagine is actually what you can get. Because by applying such kind of platform technology on your manufacturing, you can save a lot of trouble. You can meet fewer challenges throughout the entire process. Developing such kind of GMP process, actually, it's causing a lot of time and money, which, for example, investors don't want to see. So let me give you a few examples on how VectorBuilder was developing CGT together with our clients. And of course, confidential information is hidden. So the first case study is a project-specific lentivirus purification optimization. So with a very common purification strategy, we have been experiencing a great loss of virus for this project because the requirement from this customer is slightly different from the usual lentivirus design. By adjusting the purification process from the perspectives of materials, equipment, parameters, and even switching certain steps, we have reached a high HCP whole cell protein, whole cell DNA removal in our GMP batch. The recovery actually falls into the 10 to 30% range, which really based on the final titer of lentivirus that the customer wants. And the purified lentivirus yield actually is more than 1 times 10 to the 10th transductive units per liter of harvest. In our second case study, we have been doing a lot of backbone optimization for plasmid. Plasmid, it can be used as vaccine, template for IVT mRNA, or it can be used in transient transfection for virus production. So we find out that for plasmid backbone, actually it greatly impact the manufacturability of such kind of plasmid materials. And at the end of the day, it links up to your yield of your plasmid, and also it links to how easy you can remove the impurities. Like, for example, when you have higher plasmid yield per E. coli cells, it's just easier to remove the impurities when you want to retrieve more of your product. And of course, it then links to your production scale. You can achieve a certain amount of plasmid within, for example, just a 10-liter fermenter. You don't need to go higher, like bigger scale, to save your money and time. Then the last case study here is actually a bit challenging. It's an oversized lentivirus project. So we didn't only provide process development, we actually start developing a new packaging system plus process development for this lentiviral drug design. So it is oversized cassette, which is roughly 1.3 kilobase over the regular lentivirus genome size. So before the optimization, we get a very low functional titer from the harvest, and the highest positive ratio of the gene-modified cell only reaches about 16.5%. That is the highest that the customer could get. So after we gave development a new packaging system and a little bit modification on the transfer plasmid without touching the wild-type lentivirus genome, we actually achieved a much higher, almost 5 times higher functional titer with the same scale of lentivirus production, and the highest positive ratio of the gene-modified cells actually reaches higher than 40% after we do the whole development and process development project. So after showing such a case, let me introduce, in a wider angle of our CDMO service. So for our CDMO service, we have great capabilities, more than 10 GMP suites, and the current facility is actually about 100,000 square feet. We are capable of phase 1 to 3 manufacturing projects. We even have individual process development laboratories and also quality control laboratories. We are able to provide reasonable, phase-appropriate, cost-efficient products and solutions for different stages in your pipeline development. For example, research grade for drug discovery, GMP-like virus or plasmid is actually manufactured under a very similar way to the GMP grade materials, and is very suitable for preclinical study, toxicology study, and so on. So after you get enough safety or efficacy data, we can even help you to prepare IND application documents and proceed to the GMP batch manufacturing for your clinical trials. So for some very phenomenal factors, let me introduce the technology platform. For plasmid, we have actually provided more than 150 batches of manufacturing campaign. We have a very flexible custom scale from 10 milligram to 5 gram. And I'm actually very proud to present that we have a very nice process development platform to boost your plasmid DNA yield. So after a series of optimization, for a lot of plasmid, our clients' plasmid, actually the yield has boosted up to twice or even 3 times of the original yield. And let me present our latest technology, the proprietary MiniVac platform. So MiniVac is a miniaturized backbone that is incorporated into your plasmid design, so you don't have to use antibiotic screening anymore. And in terms of the fermentation, we don't use any supplement and then keep doing this natural screening along the way. So to pair with mini hosts, we can actually achieve a much higher yield for your MiniVac plasmid. And we have been also verifying that such kind of MiniVac backbone plasmid is actually showing very promising result in different kind of vector systems, such as viral vector packaging, for transfection efficiency, for example, transposon systems such as Sleeping Beauty or Piggyback. So next, let me show you the lentivirus CDMO technology platform. So currently, we are adopting the most common and the most approved generation, the third generation of pseudotype lentivirus packaging system. So that it's easier to get approved by regulatory agency all over the world. FDA, EMA, and even Australia or Japanese government, Korean government. It's more aware of the safety of this third-gen lentivirus by removing, for example, the TAT, the TAT genes. So for such kind of lentivirus, we can provide a flexible scale from 10 to the 10th to 10 to the 12th TU, and we have already manufactured more than 15 batches of lentivirus to be used in preclinical and clinical trials. So we also have massive lentiviral PD experience to facilitate the large-scale production in suspension system and also in adherent system. So currently, we have 2 FDA-approved clinical trials that our customer contracted us to manufacture the lentivirus drug substance, and we have actually 3 more ongoing IND campaign. And with our proprietary lentiviral packaging system, we are confident to say that the performance of the lentiviral vector will be higher than a common design from the literature. And the next one is the adeno-associated virus, AAV. For AAV, our flexible range will be from 10 to the 14th to the 10 to the 17th GC. We have already manufactured 22 batches of AAV. And of course, again, like we can use suspension or adherent system for the cell culture to produce such kind of AAV vectors. And of course, for AAV vectors, there are a lot of serotypes available. And we have actually tried to troubleshoot the low yield issue for a lot of different serotypes, such as 2.7 MA, AAV4, AAV6.2, PHPEB. They are actually very famous for their low yield. And we know that we have been optimizing the packaging system inside VB. And together with our process platform, we can really boost the viral yield and bring the cost and the total manufacturing campaign time down to a point that our customer can actually make the investor happy. So far, there is 1 successful human use of our AAV vector, and there are 2 more ongoing IND campaigns that is in the IND stage, to communicate with the regulatory agency. So to support some of the drug developers that are building their own facilities, but they don't want to expand too much into the plasmid preparation, they only want to do the manufacturing of the viral vector themselves. VectorBuilder is providing off-the-shelf GMP packaging plasmid. So for lentivirus, it is the GECPO, RAF, VSVG plasmid. For AAV, there is the Halbert plasmid and also RAPCap, and currently we provide serotype 2 and serotype 9. So a few highlights for our off-the-shelf packaging plasmid is that, of course, it is kanamycin resistance to meet the regulatory requirement that no beta-lactam antibiotics can be used. And for research stage and also for clinical stage, it will be royalty-free up until you commercialize, then we can negotiate the license issue. So, of course, our GMP-like and GMP-grade packaging plasmid is of high quality so that it can help you justify the safety and efficacy of your drug. So, last but not least, why should you talk to VectorBuilder? So, VectorBuilder have been accumulating so much experience in vector design and the production of such vector. we really want to transform our know-how to designing your drug, develop your drug, manufacturing your drug. And of course, accidents always happen. And our know-how can actually become your resource to troubleshoot and to support your product development. Because at the end of the day, your time, the progress of your drug pipeline, and your budget are valuable. And we want to provide such a platform that we can help you and really work with you and make this drug happen. Thank you very much. If you have any questions, I think next we have a Q&A sections. You can, feel free to ask me any questions. If you are maybe a little bit shy, you can contact our service email, which is shown on the screen for more information. Thank you very much. 감사합니다.